So if you are at Emory University or healthcare employee and would like to receive CME credit for attending today, the access code today is 806186 and can be found in the chat feature at the bottom of your screen for virtual attendees. If you have any issues with the webinar or the CME login, please send Janelle Taylor an email or drop a note via the chat feature. This morning, we are pleased to welcome Dr. Ted Lawrence. Dr. Lawrence is a professor of radiation oncology at the University of Michigan, where he was chair of the department for 25 years, 16 of which I was on faculty there and was his deputy chair for quite some time. He's now co-director of the Translational and Clinical Research Program of the University of Michigan's Rogel Cancer Center and an editor of multiple books, journals, author of countless papers. He's been elected to the AAP, the National Academy of Medicine. He's been president of basically everything: ASTRO, the Radiation Oncology Institute, SCAROP, which is the Society of Chairs of Radiation Oncology, and chair of the National Cancer Institute's Board of Scientific Counselors. He was a member of the board of directors of ASCO, founding chair of Radiation Sciences and Medicine as a working group of AACR, and has been awarded the gold medal of both ASTRO and the Israeli Society of Clinical Oncology, an ASCO Statesman Award, RSNA's Outstanding Researcher Award. And as Ted, he didn't say this, that was going to take up his entire talk, but I could just go through his many accolades and actually just use up his time. What I'd rather do is tell you something a little bit more personal about him, because he's going to talk to you about his very important research. And that is that Ted is as well known to those of us who have worked with him for his mentorship of individuals to become leaders in the field as he is for the quality of science. And you just heard about how impressive the science is. So he is probably the department chair who has mentored more people who became department chairs than any other person in this country. So I heard recently, and it's made me a little bit sad, because somebody told me that Harvard actually had more department chairs in radiation oncology. Harvard's the top one, and Michigan is number 2. And somebody emailed me and said, hey, you count for both of them. And I was like, can I just count for Michigan? Because it really is, Harvard had many people and many hospitals and, multiple chairs who led to that finding. But Michigan really had one, and that was Ted Lawrence. And he really has been such an inspiration to all of us and such a leader within our field, centering our field and being able to communicate about things that are so important, including molecularly targeted radiosensitization, which builds on his interest in the laboratory, that are focused on molecularly and immunologically targeted radiosensitizers. Literally every clinician in our department ended up with some kind of work in this space. I got an IND and ran a phase 1 and now have a phase 2 clinical trial in SWOG, in the National Cancer Institute Cooperative Group System, on radiosensitization using a PARP inhibitor. So literally every single one of us had something to do with this. And I see this as a potential opportunity for us to learn how we can leverage the strengths within our department to submit more multi-investigator grants. There's already a foundation and a history here of doing that. And so with that, I am so pleased to invite Dr. Lawrence to present to us today.
Thank you very much, Dr. Jagsy. That was, I wish my mom had been here to hear that. Thank you very much. Really great to be here. I've had lovely visits so far. And my first visit here to Emory, and it's just been great. You have a wonderful facility here and terrific faculty. It's been a terrific visit so far. And thank you all for being here. It's 7:30, kind of early-ish, so thank you. So disclosures, we miss Dr. Jagsy a lot at the University of Michigan. I would say all the things back to her that she just said about me. Dr. Jagsy is a phenomenon. I don't know how else to characterize her work ethic, insights. The only arguments we've ever had about various issues, she's actually turned into papers. And so, and you didn't acknowledge me, at least on one of them. So my advice to all of you is, which I got from Anthony Zeitman, who's a well-known radiation oncologist in the Harvard system, is don't argue with Dr. Jagsy. Resistance is futile. I did want to show one picture from a number of years ago. This is I think 2018, something like that, when Dr. Jagsy became an Astro Fellow. Very, very proud of that. And Jim Heyman worked very closely with her and with us. So I want to now try to take a larger view beyond even my own work and talk about the work of a lot of people today with the concept of rational hypothesis for improving outcomes in radiation therapy. So my claim is that solid tumors cannot be cured without eliminating the primary tumor. I think even medical oncologists would believe that, right? Those of you who are in the Ethernet. With a few exceptions, systemic therapy does not cure gross disease, but can cure microscopic disease. We have great evidence of that. So radiation therapy, if you want to take a very large view, it has the potential to eliminate the primary tumor in at least two different ways. One is to improve delivery, permitting normal tissue sparing and dose escalation. That is technical improvements. And technical improvements have been very important in the field. And then the use of agents that selectively make tumors more sensitive to radiation. And then, of course, using both approaches. So I'm not going to deal much with technical aspects, and we'll talk a little bit about that in a moment. I'm going to focus more on the biological side. But there's no reason that these both can't be used together. And in fact, for most radiosensitization approaches where there could be some more normal tissue toxicity, using the best approaches technically permit you to do more by conforming the fields to the tumor itself. So dose escalation can improve outcome. And I'm just showing you an example of work from Michelle Merzois and her team at the University of Michigan, following on the work of Avi Eisbrook, who started this protocol. And this was published now in Cancer Research a couple of years ago. In a phase 2 randomized trial at 2 years, there were 15 local regional failures in a standard arm for the treatment of locally advanced head and neck cancer patients, and only 7 local regional failures in the RT boost arm. So these are patients who had treatment intensified based on advanced imaging to the aggressive parts of their tumor in a randomized phase 2 trial. The cumulative incidence local regional failure was significantly improved with the addition of the radiation boost after adjusting for the same prognostic variables. So I'm just giving you one example from our work. There are other examples in which dose intensification can improve local control. But it doesn't always work. And this is the attempt to dose escalate in diffuse midline gliomas was not successful. This was a trial that was run by Larry Kuhn, who passed away a number of years ago at St. Jude. And the trial was trying to intensify dose to DIPG using, I think it was 1.17 gray BID up to 70 and a bit versus the standard. And as you can tell by the survival, really dose intensification in this setting didn't produce any benefit. And maybe the best known trial in our field right now in which dose escalation didn't benefit was in stage 3 non-small cell lung cancer. And this is RTOG 0617. I think probably everybody in our field knows this study, which compared standard dose 60 gray versus high dose 74 gray radiation with concurrent chemotherapy for stage 3 non-small cell lung cancer. And the lettering is a little bit small on that, but for those of you who aren't on this, don't know this study, unfortunately, the control line is the upper line and the experimental line is the lower line. So there's actually a survival decrement. There's been a tremendous amount of study as to why this might have occurred. Could be cardiac dose, could be other reasons. But the reason I show it is that, this set of slides is that technical advances are important. They can produce improvements, but we can't uniformly count on dose escalation to improve outcome. So adding chemotherapy to radiation improves outcome. I'm not going to go into this all the examples here, but I think as I look back at my career over the last 20, 25 years, this is in my opinion, probably the most important finding of all in oncology, which is that using chemotherapy alone produces one outcome, radiation alone another, but combination chemotherapy with radiation therapy, when done appropriately, can produce improved outcome in all of these settings. And we can argue about pancreatic cancer some other day. My main interests clinically are in GI and brain, but I think I could defend pretty much all of these, and I think you'd agree with me. But where do we go from here? Can we improve the results of combining chemotherapy with radiation therapy by using agents that are targeted to the pathways that cause radiation resistance? And the hope would be that this would be more selective. We hypothesize that molecularly targeted agents that selectively radiosensitize tumors and stimulate a systemic immune response, if that's possible, will improve the outcome of treatment for patients with locally advanced cancers, which is probably roughly a third of all cancer patients have locally advanced cancers. So we're now testing this hypothesis in a specific way in our radiosensitization spore that just was funded starting last year. So our hypothesis is being tested through 3 projects, each of which carries out preclinical studies leading to a clinical trial approaching radiosensitization from a different angle. And this was something that, as we all got together, I think everyone got excited about, that there are different ways of trying to overcome radiation resistance, and we could potentially use individual organ sites to test these different concepts. So project 1 is using olaparib with radiation therapy and durvalumab for patients with unresectable pancreatic cancer. And here we are trying to make a cold cancer, pancreatic cancer, and immune stimulated. Project 2 combines mycophenolate mofetil, MMF, with radiation therapy and temozolomide for patients with glioblastoma. So this is trying to overcome metabolic resistance, which I'll show you evidence of. And then the third project combines a CDK4/6 inhibitor, abemaciclib, with radiation therapy for patients with locally advanced ER-positive breast cancer. And the focus on that project is on cell cycle and on the DNA damage response. So we thought these were 3 very important ways in which tumors resist treatment, and that by using targeted therapies, we could potentially overcome that resistance. And what I want to do is show you the preclinical rationale for each of these and show you the clinical trials that have started to emerge from that with the hope that this is going to give us some improvement. So we tried to structure this in a way that would test our hypotheses. So the goals for the projects, or the primary aims, of course, to understand the biology underlying radiosensitization in vitro and in vivo, but then at the same time to try to translate that into clinical trials to determine the maximum safe dose of the agent under study. And then probably most importantly, even though they're secondary aims of the clinical trials, we want to estimate the response. We were having this discussion last night at dinner that whenever you do a phase 1B sort of study, you still want to know at the end of the study, is this worth pursuing? Even if the test is tolerance and feasibility, if at the end of the study you don't know where to go, then you haven't gone in the right place. And then assess the preclinical hypothesis in the patient. I think this is such an important factor, and I'm sure you all agree that you run a trial and not every trial is going to work, right? So you want to know at the end, did you hit the target? And if was the target relevant? Because there are at least 2 reasons why a trial might not be successful. One is that it looked like it was a good target and it wasn't. Or the other is the target was that you hit the target and it wasn't effective. Or the other is that it was never a good target in the first place, and you want to know which it was. So this first project is using PARP inhibition with radiation to sensitize HR-proficient pancreatic cancer to immunotherapy. So I want to talk about the cartoon for a moment. But the concept here is not, we often think about PARP inhibitors in the context of synthetic lethality. And this is not thinking about PARP inhibitors this way. This is thinking about PARP inhibitors as radiation sensitizers, as blocking DNA repair. And so the hope, the model, working model that we have for this concept is that we're combining the clinical trial didn't turn out to be SBRT. We'll talk about that, SBRT-ish, with a PARP inhibitor, with the concept that radiation alone might not produce enough DNA damage in order to stimulate the immune response. And there have been several studies now that have suggested that radiation alone might not be sufficient. So by adding a PARP inhibitor and increasing DNA damage, the hope would be that you would get a greater increase in cytosolic DNA, that would stimulate the pattern recognition receptors, leading to type 1 interferon. And then that would lead to further tumor effects and also host effects that have to be taken into account. And then hopefully with therapeutic efficacy by adding a PD-L1 inhibitor. And I'll show you that in a moment. So the aims of this is to elucidate novel mechanisms of innate immune modulation, pattern recognition receptors, and their type 1 interferon-dependent phenotypic consequences. Then determine the therapeutic benefit, immune contribution, and molecular endpoints of combined therapy with a PARP inhibitor and immune checkpoint blockade, and then conduct a clinical trial with radiation, durvalumab, and olaparib. So what's the preclinical basis for doing this? Well, PARP inhibition can potentiate radiation-induced type 1 interferon. And this is shown here in this reporter system, which lights up, the GFP lights up with the interferon reporter. And with increasing, this is concentrations of olaparib with or without radiation. And you can see that the reporter lights up much, much greater in the presence of the combination of the two. These are two immune competent systems in mice, in genetic systems, KPC2 and MT4. But again, both show in cultured cells that when you radiate in the presence of olaparib, you get much greater stimulation of a type 1 interferon response. So we know that PARP1 inhibition, at least in these model systems, can stimulate a type 1 interferon response. So PARP inhibition also enhances interferon-stimulated gene response. It works down the pathway. And so what's being shown here is a number of the different cytokines. I can get my glasses there so you'll read that. CXCL9 and 10, and then these are MHC1 and PD1 are all stimulated by PARP1 inhibition with olaparib. But then if one looks at this whole panoply of what's occurred, you can see that there are potential negative effects as well as positive effects of having done this stimulation. In the case of negative effects with PD-L1 expression, that could actually produce immune invasion, right? If the tumor cells are showing PD-L1, they might avoid being targeted by the immune system. Whereas all these are positive effects: antigen presentation, chemokine production, and increased CD8 T cells. That will help in the anti-tumor response. So I think it seems obvious where this would go from here, right? That we would want to use something like durvalumab, something that would attack the PD-L1 expression and remove that as a protective mechanism for the tumor cell. Now, this would suggest that this whole approach would require an immune competent system. And so, and this is work that, again, Meredith Morgan in our department has been leading with Visa High, who's a medical oncologist on the clinical trial. And here, what she's shown here is comparing wild type mice to immune deficient mice, looking at control, olaparib, radiation, and then the combination. And this is now an experiment in vivo study. And if you can compare these two curves, you can see that olaparib is barely a radiation sensitizer in animals that are immune deficient. But in the wild type animals that have an intact immune system, olaparib plus radiation is a much more effective combination. And I think some of some of what was in the previous slide is illustrated here. You do get an increase in PD-L1 with this combination. So this motivates the use. You can see on this Western blot, PD-L1 is up, and the combination of radiation and olaparib, and shown here on the bar graph. And so this does motivate a clinical trial concept, adding olaparib to radiation to get increased sensitization, increased DNA damage, but then durvalumab to potentially overcome the PD-L1 response. So now PD-L1 inhibition, to test that in the laboratory, it looks like it does potentiate PARP1 radiosensitization. Now there are a lot of lines. Here's the experimental design of radiation. We can talk a lot about whether 8 gray is a magic dose in the laboratory, and there's a lot of discussion from Dr. Fermenti's work on this, but I'm not going to go into that at this moment. But we get a combination of olaparib given by oral gavage in these animals, plus PD-L1 plus 8 gray doses of radiation. And here are all the controls on tumor volume that are all clustered for the controls. But the key issue is out here when one adds the PD-L1 inhibitor plus olaparib plus radiation, you get the best response, combining all of these together in one concept, blocking the immune protection that PD-L1 might provide. Now, why is this occurring? Well, the evidence we have so far suggests that host CD8 T cells and type 1 interferon are required for treatment efficacy. And what's being shown in these slides are, again, tumor volume in intact animal system. And we have olaparib plus radiation plus anti-PD-L1 plus anti-CD8. So when you take out CD8 cells, you compare these two, you take out the CD8s, you do get some effect, but the effect is partly mitigated by not having CD8 T cells. And the same concept in this experiment, this difference is showing when one removes the type 1 interferon by having animals in the host animal, by using animals that don't have the type 1 interferon response. So you need all of these together, which we hope are going to occur in patients to produce the best response in this system. So here's the clinical trial that is running based on some of these concepts. This is for locally advanced unresectable pancreatic cancer on first-line chemotherapy for at least 16 weeks without progression. We had a lot of debate about this, but I think the standard now in pancreatic cancer for locally advanced disease is to give people chemotherapy up front. They're not going to respond at all. I think the chances of long-lasting survival are relatively small. So we're trying to pick a subset of patients who might benefit from intensive local therapy. And then this was I digress for a second. This was a trial that was constructed, I'm debating whether to say this constructed with CTEP or against CTEP. I don't know how many of you have worked with CTEP. It's you've heard the expression that a camel is a horse made by committee. This is I'll just stop there. I may start crying in front of you if I pursue this too much further. But anyway, this was what we finally were able to negotiate. It's an 18-patient trial. It really should have been 30 patients. I'm not sure about the olaparib and durva with a requirement for the cycle 1 to show that was tolerable, and so far it has been. And then this is the meat of the study. And we actually wanted to use SBRT, but we've wound up using 39 gray and 3 gray fractions. This is one of those having a beer at a bar discussions, isn't it, about how we got here? But the hope is going to be that we're capturing the main concepts of the preclinical work by adding olaparib as a sensitizer, durva to overcome the PD-L1 response, and focused radiation to try to deliver DNA damage along with the olaparib sensitizer to produce the response. Primary objective, of course, is to determine the maximum tolerated dose or the proposed phase 2 dose with fixed and we picked durvalumab and radiation sort of a unit. We thought that would be safe, and the olaparib is dose escalated. We're already at level 2 for olaparib. I think probably the dose that we're going to be at. And it is a multi-center trial. I could talk more about it. I'm going to leave it as it is. I want to just throw in one slide on using this very general approach in trying to treat a diffuse midline glioma. This is actually a different approach to try to determine the ability of PARP1 inhibition, selective PARP1 inhibitor. Olaparib is a PARP1, PARP2 inhibitor, and there's some evidence that PARP1 inhibition is more selective anti-tumor, and PARP2 may have more of the toxicity side to treatment. But this is emphasizing looking at NK cells and whether NK cells can be mobilized in DIPG or diffuse midline glioma. So in ones that you can see there's determined PARP1 inhibition and radiation to stimulate the innate immune response in these DIPG cells and determine the therapeutic efficacy and immune contribution in syngeneic and autochthonous DIPG mouse model. So this AZD9574 is a PARP1 selective agent. These are the model system showing the implantation of the orthotopic implantation model for DIPG. It's a very tricky model that our folks have been able to master. And then we do have not a huge effect, but it's the beginning of an effect here by using this selective PARP1 inhibitor in the setting. And then these panels are showing that we're getting a higher fraction of NK cells, and these NK cells are activated. They're TNF-alpha positive activated NK cells. So we're hoping that there may be something we can do for DIPG. University of Michigan has a big push now in DIPG. Our football coach, grandson, unfortunately died of this illness. And he is one of these shakers and movers and has raised well over $50 million to support research in DIPG. So we're trying to leverage that interest with our ability to contribute to that. Now I want to turn to high-grade gliomas, the work of Dan Wall and his team. And this is the project that I'm most involved in personally. And what Dan has discovered is that high purine levels correlate with radioresistance. And he began this experiment with looking at this concept by looking, I think there are 23 cell lines there, and determining their radiation sensitivity, and then determining looking at gene set enrichment analyses originally, trying to figure out what's different about these 23 different cell lines, where some of them are quite sensitive. This is the mean inactivation dose. It's a measure of radiation sensitivity. So you can see there's a spectrum of sensitivities across all these lines. And so 3 of the top 10 associated gene sets with radiation sensitivity were related to oxidative ATP production. That's not a big surprise. Oxidative damage is a major, it's well known that this is involved in radiation sensitivity. There weren't any metabolic gene sets for the gene sets, but if you did a direct metabolic analysis, it showed purine depletion was associated with radiation sensitivity. And that's shown here. All these bars are on the depletion side. So purine depletion was associated with radiation sensitivity. I do have to give you a vignette for all the young people in the room. They worked months to create all these samples, these metabolic samples, and something went wrong in the shipping, and all of them were destroyed. And we all looked at each other and said, that was $50,000 down the drain. Do we do this again? And sometimes there are really great reasons for being chair. I was able to scrape together the money, and it turned into this whole project. So sometimes there's a happy ending to a sad start. So why does purine synthesis, or can purine synthesis inhibit radiosensitization? So here's the pathways that are involved in purine synthesis, GTP and ATP. So it was clear that there was a correlation, but the big question, of course, is it just a cause or correlation? And so in this experiment, going now to the animal studies, the luciferase positive radioresistant GBM38 patient-derived xenografts we got from Jan Sarkaria at the Mayo Clinic were orthotopically implanted. The mice were randomized to radiation MMF or MMF plus RT. So MMF acts, this is CellCept. This is a drug that's been used in tens of thousands of patients as an immunosuppressive agent, possibly through GTP depletion. Maybe some in the audience knows that. But the idea is that MPA or MMF inhibits this pathway. So you can imagine if the cells were getting their guanine nucleotides in particular through salvage, this wouldn't work, right? The salvage pathway would come in beyond where MMF would be doing the blocking. MMF would only work if it's new synthesis. But in fact, when MMF was administered, as is outlined here, and the mice were treated with luciferin to light up their tumors, you can see in the combination treatment, this examples of the mice. Here's the controls, radiation, MMF alone in the combination. And then also, these are the bioluminescence and the survival were all improved by adding MMF. So this was a suggestion that inhibiting guanine nucleotides would actually produce a positive result. Now, why is this happening? The purpose of the spore is more focused on translating findings into the clinic, not so much in diving deep into mechanism, but this is a really beautiful study that Weha Zhou and in Dan's lab, and now she has her own laboratory, but in the work that he was helping to supervise, asking what is the mechanism by which this is happening? Because we initially thought it makes sense that these were just DNA building blocks, that you've damaged DNA, you deplete nucleotides, the cell just doesn't have nucleotides to repair their DNA damage. But it turned out to be guanine nucleotides. And why would it just be guanine nucleotides we're doing this, not all nucleotides? And so what Dan discovered in this approach is that GTP, but not other nucleotides, regulate the activity of RAC1, a guanine nucleotide binding protein, which promotes the dephosphorylation of serine 323 in ABI1 by protein phosphatase 5. So it's dephosphorylated ABI1 turns out to be involved in DNA repair. So this starts with 17,000 different phosphorylation sites throughout the cell. And so the question, this phosphoproteomic approach was asking, what in the cell, what in these gliomas were induced by DNA damage, augmented by G supplementation, and blocked by GTP depletion, by MPAs? You want all three of those things. And ABI1 is the one that popped up by when it's strongly dephosphorylated. That's this dot here, 2 logs of dephosphorylation. That's what led to the ability to repair DNA damage. So ABI1, dephosphorylated ABI1 activates non-homologous end joining. So this was a unique finding and not known before. And in patients in mouse models of glioblastoma, RAC1 and dephosphorylated ABI1 mediate DNA repair and resistance to standard of care genotoxic treatment. So we're pretty excited about that mechanistic study. So here's the actual SPORE project. I've already mentioned this part of the activity before, which is that when you have low levels, high purine pools, DNA repair, cell survival, radiation resistance, and you have low levels, radiation sensitivity, and that the hope is that MMF can convert the resistant into the sensitive. And I guess our hope is that this A plus G thing doesn't happen. And then more recently, what we've been looking at is how the astrocytes handle the precursors that make the guanine nucleotides. And so under normal conditions, astrocytes and organoids make substrates for the neurotransmitters. That's what the brain wants to do for a living, right? Is to make neurotransmitters and communicate with the body. But what tumors do is they manage to hijack those glutamine and serine and use them for purine synthesis and other nucleotide synthesis and use them to grow as opposed to use them to do good things like communicate. And so the question would be, can one interrupt these pathways by using agents to block this? And do these things actually happen in patients, not just in animals? So to turn metabolic mechanisms, measure de novo purine synthesis using in vivo stable isotope tracing, and then perform a phase 1 trial combining MMF with standard therapy in patients with glioblastoma. So working on these pathways to try to prevent tumors from stealing these glutamine and serine into making what's going to permit them to grow as opposed to communicate. So the team, along with neurosurgery and anesthesia, it's a large team of people, have been able to do isotope tracing in human beings. And so the approach to this was to the glucose is made from carbon-13. So carbon-13 is a heavy, non-radioactive isotope of carbon, and you can trace the C13s through the metabolic pathway. So the patients are infused prior to surgery with glucose, with C13 labeled glucose, and achieve steady state levels over maybe a 3-hour period. And this is the arterial glucose. And then the surgery occurs, and what they looked at was the enhancing tumor, non-enhancing tumor appearing, and normal brain. And so what's shown in these 2 panels, the key result is that if you look at the GBM enhancing and non-enhancing behave about the same across both these panels. But the question is, what's being made from the infused glucose? And so for pyruvate, citrate, and UDP glucose, you can see the normal brain is using these more than the tumor. So these are the normal brain panels, and these are the tumor panels. But when it comes to GMP and GTP, the guanine nucleotides we were just talking about, the tumor is pulling them up from the plasma, from the serum, much more than the normal brain. So this, again, would give you hope that if you could block that uptake, if you could block the ability of the tumors to take these up, using something like using MMF, that might deplete those levels and then permit you to increase the sensitivity of the tumor to radiation and temozolomide. So here's the protocol that, and this actually, this newly diagnosed GBM phase 1 trial has actually already completed accrual. It was an easy trial to accrue to. We've already accrued the 30 patients, which is the right design. And it uses a time to event continual reassessment model, which we can talk about it at some point if people would like to. But the basic idea is that patients get a week of MMF, then they get MMF, then they get temozolomide and radiation, that's standard treatment, with MMF at the same time. Then they get an assessment for DLT, and then they continue to get temozolomide. Again, that's the standard treatment with MMF added to it. And so far, we haven't fully analyzed results. The trial's only been running for a couple of years now. I know the SPORE only got funded. You know the way it goes, right? You have to do at least a third of the work, half the work before you get your grant funded. So we did a lot of this before it actually got started. But the initial results have been very promising, and we've been working with folks at Northwestern, where there's a randomized trial concept that Alliance has approved, the Alliance, and they're going to hopefully take this to a randomized phase 3 trial. And then we want to look at differential serine metabolism in GBM. You could imagine a serine restricted diet. In the animal studies I'm not showing you here, we have some evidence that by restricting the diet, you also can slow tumor growth by restricting them from these guanine nucleotides. And then hopefully taking that concept to a clinical trial. So finally, I want to turn to the third project, which is the high recurrence rate of greater than 3 positive node ER positive breast cancer. So Reshma, I'm a little nervous talking about breast cancer in front of you. You're so much more expert than I am. But so if I make a bad mistake, you can raise your hand and you can fix me, you correct me. But this is a project being run by Corey Spears, who's now moved to Case Western. And in this project, patients with estrogen receptor positive, ER positive, multinode positive breast cancer do have a high risk of local recurrence, a local regional recurrence. And then cyclin-dependent CDK4/6 inhibitors are approved for metastatic patients with ER positive breast cancer. And recent clinical trials have explored the use of these inhibitors in patients with non-metastatic ER positive breast cancer. And the question is, can you use these with radiation? And will that possibly improve local control? So the CDK4/6 inhibitors, again, many in the room understand these, but just very quickly, as one goes from G1 to S, you have to have activated cyclin D CDK4/6, that complex, and that complex, when it's moving through the cell cycle, will phosphorylate RB. RB will fall off E2F, and E2F can then lead to transcription and moving the cell through the cell cycle. And so what we had all thought when Corey started this work was that cells would be synchronized by a CDK4/6 inhibitor at the G1S boundary. The radiobiologist in the room will know that's a relatively sensitive phase in the cell cycle. And so we thought, that's great. It's going to redistribute cells into this boundary and that's going to produce sensitization. We were excited about that. And in fact, it did produce sensitization. Corey has used all 3 inhibitors, palbo, ribo, and abema, and they all produce essentially the same results. There's some detailed differences. But in MCF7, ER positive cell line, and TD47, D ER positive, you can see these are all radiation cell survival curves. This is log survivin fraction versus dose of radiation. And increasing concentrations of the CDK4/6 inhibitors produces increasing radiosensitization. That's all good. That's all what we had hoped for. And what are shown in these panels are just the 2 gray radiosensitization, a surviving fraction of the 2 gray, which just mimics the full cell survival curve. And MCF10As, which are normal-ish immortal cells, are not affected by this. So we were very happy by this. And then we wanted to try to understand the mechanism by which this is occurring. So AIM-1 are the in vitro studies, AIM-2 in vivo to try to show if this actually works in the animals, and AIM-3 would be hopefully taking this to a clinical trial. So this was, again, we were eager to see if this reproduced itself in animal studies, and palbo treatment leads to radiosensitization of ER-positive breast cancer cells in vivo. Here's the outline of the study. The we're using an interesting model system of trying to implant a tumor into in an intraductal way with estrogen pellets to try to produce a more relevant animal model. And then animals were radiated. Down here they got radiation, or there we go. A vehicle alone, palbo, radiation, the combination, and again, the combination of palbo and radiation was better than any of the other conditions. And again, in these xenografts, this is percent of tumors that have not doubled in size. And so again, vehicle, they're doubling, but in the combination treatment, there was a big difference in the median time to tumor volume doubling. This is all what we, you hope for a certain result. And this is where it got interesting. I think it got interesting to everybody, which is that the focus in this area has been more on the ER positivity, but what Corey's work is now focused more on is the RB dependence of this phenomenon. And maybe from that the CDK4/6 slide that I showed you, that should have been anticipated. But in these two conditions, this is MCF7 and MDA-MB231, and he knocked down RB in these two panels, these two bars, but under the control conditions, this is the these are the control RNAs. So again, in RB competent, this is these are looking at RAD51 foci. So if you have RB present, the combination treatment leads to a lot fewer RAD51 foci, suggesting there's less homologous recombination repair going on in these cells. In both of these conditions, when you take out RB, there's no difference here, no difference here. But then this is really the interesting panel to me, because you can do the pre-exposure for just an hour or two and get the full sensitization. But during the this is showing cell cycle redistribution, percent of cells in G1. And you can see during the first 6 hours, nothing much is changing. And during these first 6 hours, nothing much is changing. So it's not cell cycle. The cell cycle isn't changing, but we're getting sensitization. And so now I think the focus of these studies is going to be more on detail. What's the role of RB in homologous recombination repair? There are some reports of that. I'm not sure that's entirely novel, but that's going to send us down a different pathway toward understanding the mechanism. And here's the clinical trial that's underway, enrolling patients with the inclusion criteria are 1 to 3 lymph nodes positive in T3, 1 to 3 lymph nodes positive in G3, or greater than 3 nodes. And these patients are going to get their standard radiation with ribo. And we have an additional arm to this, working with Lynn Henry, who's very interested in patient-reported outcomes. We thought in particular for an adjuvant study, these are all women with resected disease, particularly important to understand what the toxicity is always important. But in people with life-threatening illnesses, you're willing to subject somebody to a little more toxicity. Excuse me. In an adjuvant setting, I think you have to be unusually careful and making sure you're not degrading somebody's life for a percentage of improvement in outcome. Okay, so in summary, locally advanced cancers cause substantial morbidity and mortality. Systemic therapy alone does not typically cure the primary tumor. Improvements in planning and delivery of radiation have improved outcomes. I didn't emphasize the technical things today. My other life that I thought about talking to you about, which is treating hepatocellular cancer with conformal radiation. The focus is much more on technical issues and on modeling the dose response effect. So I think I'm still interested in those, but I thought this would be a little different for you to hear about today. And we propose to combine state-of-the-art radiation planning and delivery with novel biologically based treatments aimed at the drivers of radiation resistance will hopefully result in some new cancer cures. This is the SPORE team. It's 3 colleges, 7 departments, 30 investigators. What do they say, it takes a village. I think one of the most exciting parts of this for me was just seeing this team work together. And it's really thrilling to see everybody pulling together to do something that none of us could do individually. And finally, that's my license plate. Yeah, I love radiation therapy. I think it's a fantastic field. I think with improved technical abilities and improved understanding of biology, how to integrate biology with radiation, there's a great future in the field and a lot of great work to do. So thanks for your attention. Thanks for being here early. to hear this, and I hope stimulate a little thought.
Thank you, Dr. Lawrence. So if you have questions online, please use the Q&A feature at the bottom of your screen. And while we wait for questions, I just wanted to mention that next week there will be no grand rounds, but please do keep an eye on the Winship grand rounds calendar on the Winship website to see where upcoming grand rounds are going to occur. So we've already got a question in the audience from Dr. Ramalingam.
Thank you. Yeah, so it's really a good question, isn't it? It's the results have been a little mixed, haven't they? I think there's strong evidence, not just from our group, many groups, well, before our group, that PD-L1 is expression increases when one does radiation. And then the question is there a way to overcome that, and can we use that? And so there was a very well publicized JCO negative trial in head and neck cancer. And in fact, our attempts to add PD-L1 inhibitors to cisplatin and radiation head and neck cancer produced a clear result. No benefit, increased toxicity. That's the study. We have about a 30-patient study that we're writing up right now. Maybe you've had a better outcome the way you've combined them. And so more toxicity, not greater efficacy. That's what we've seen. I think there was a lung cancer trial published a couple of years ago now that suggested it didn't meet its endpoint, but there was a strong suggestion of improvement. So I think it's going to turn out to be tissue type dependent, maybe even tumor dependent within tissue type. I think there's just a lot to learn, and it has not been the slam dunk everyone was hoping for. We were all thinking, this is it. We're going to overcome resistance. And then the second part of your question is there an abscopal effect, or is there a local effect or an abscopal effect? And so the animal model studies that we've done in pancreas have not been, in these model systems that I showed you just now, there's not been a strong abscopal effect, if any, in the pancreas model. But then again, I'm not sure we have not developed a model system. I think that the relevant model system for locally advanced pancreas cancer would be an occult tumor, right? It wouldn't be placing a treated tumor on one flank and an abscopal tumor on the other flank. Because with the locally advanced patients, we suspect that close to 100% of them have occult disease, but they're not showing us gross disease. So it could be occult disease response could be different from gross disease. So I think at least for our experience, the abscopal response for gross disease has not been strong. But maybe there could be an abscopal effect for microscopic disease that I don't think has been well studied yet. That's a long answer, but it's a really important question, and been a little bit disappointing, I have to say.
Great talk. So just a quick question about Project One, which I thought was very interesting also. So in that clinical trial, you guys are doing induction chemotherapy for 16 weeks. Was there concerns about how lymphotoxic that would be, given the fact that you're eventually trying to stimulate?
Yes, there was a lot of concern about that. And we are getting specimens from everybody prior to treatment, prior to our part of treatment, to the experimental part, to see if there's what we actually are dealing with in the at least the circulating immune system. And so the answer is yes, if there's a concern. A lot of balancing of how much does one deviate from standard practice? How many variables does one change from standard practice? And I think for pancreatic cancer now, it is standard practice to give 3 to 4 months up front. So we couldn't get CTEP to nudge away from that. It's a real risk. You're absolutely right, and it could be all the good guys, all the good lymphocytes are killed off by 4 cycles of chemotherapy. There's a possibility of that. Yes.
So much, wonderful presentation. Congratulations for. With respect to your GBM work, it seems like you believe that not only has radiosensitization, but chemosensitization with the temozolomide based on your study design. Have you seen much differences in responses depending on the MPFT population status?
Yes, so the temozolomide work is not ours. That's a collaboration with Northwestern. The group there, they've done all the temozolomide work. Yeah, we decided that we're radiation oncologists and we were going to focus on the radiation side. No, it's a really, because there was a desire to add it in right away, but then the question is, did it make any sense to do that? And so they have good data on the temozolomide side and it's a different mechanism from it's not a pure guanine, a GTP signaling pathway that they have. So at least the preclinical evidence suggests that there's sensitization from temozolomide alone, and the hope would be, of course, that isn't some negative interaction that's going on when you put them all together. And there was, I didn't show you, we do have animal data that the combination is also synergistic with MMF. Yes.
Just to cause for speculation, but all your work is in locally advanced primary tumors. How translatable do you think that will be to metastasis, like your pancreatic cancer metastasis? Are the same effects going to be seen now that it's in the liver?
Yeah, they might be. And of course, the TME is going to be different for the locally advanced patient compared to the metastasis. So I think that's definitely an area that should be studied, shouldn't it? With the treatment of ligamentasis being so common now. So I say it could be, but I think the TM, a lot of what we're talking about, at least for the immune response, is it could be tumor microenvironment dependent and might be different. But I think it's a great area to study. We should do that. Oh, thank you for your attention. You set me up very nicely. I was expecting 6 sleepy people this morning. This is fantastic. So I feel very, very appreciated today.
Which is, you've been so successful in obtaining funding, over 25 years of continuous NIH funding. And, you spoke a little bit about donor intent and, the what is it, Chad Tuff Foundation, right? And so it's sort of going where opportunities exist, but then also your work has shaped priorities, right? Typically spores are disease site specific, not modality focused, right? And so how do you balance going where opportunity takes you versus being intentional and following the path that you think is appropriate?
Yeah, so that's yeah. So I think that it's important that to do what you do, what we do really well, and then and deeply and thoughtfully, and then that can be used to pull other people along. I think that if all you're doing is asking other people for help, and they see you coming down the hallway and you see them sort of ducking into the door to get away from you, you'll know that that's not going to produce the collaboration you're interested in. So I think that the people I've met with here, your faculty here, are fantastic. They're bright, they're hardworking, they're innovative. So I think you have within your department the ability to move these things forward. And then the environment, of course, is crucial. It's very hard to create something from absolutely nothing. And so you want to scout for places where you need something, where there's something there, and then you can build on that something. I mean, certainly you can go to another department or to another investigator and say, we know nothing about this. Let's pool our resources and try to become a force in it. Every time I've changed topics, changed studies in my life, it's taken me at least 3 full hard years to become credible, to get papers published in decent places. So any investment in a new area is a minimum of 3 years just to even become credible. So it is that combination of what are you bringing? Are you bringing top flight in your area? What's already existing and how much you have to build on it in order to become competitive? Because I'm telling you something, science is competitive. And so in order to really play at the highest level, it's a big investment. So I don't know if I answered the question, but it's that combination of your environment and what you bring to the table. But you have it here.
Thank you. We are so honored to have you and really looking forward to the rest of the day as you meet with our faculty and our residents. Thank you again.